So in our group, we study RNA viruses. These are viruses that have their genome in the form of RNA and include many well-known pathogens such as human immunodeficiency virus or influenza virus or Ebola virus. And these are very important from a medical standpoint because they create tremendous disease worldwide. And unfortunately, we have very few vaccines or antivirals to treat infections with RNA viruses. And so our long-term goal in the lab is to try to identify new treatment options and learn as much as we can about the biology of RNA viruses. And the other reason we study these pathogens is because they have some very interesting properties. And these include the fact that they have a very small genome, and so usually they encode 5 to 10 proteins. And with these very few proteins, they're able to coordinate all the steps required to enter into the cell, to replicate themselves, and to transfer from one host to the next. And the only way to accomplish this is by being very efficient at interfacing with the cellular machinery. And so by studying how they are able to accomplish all these tasks, we can learn both about the virus and about how the cell works. And the second interesting aspect of RNA viruses is that they have the highest mutation rate in nature, which gives them the highest evolutionary capacity in nature. And that lets them escape the immune system and evolve to infect new hosts and rapidly become resistant to antivirals. So we are focusing on two aspects in the lab right now. The first one is how RNA viruses interact with a group of proteins in the cell called chaperones. In the past, we've shown that viruses rely on chaperones for their replication and that you can inhibit chaperones, and this is an antiviral approach. And so what we're doing is trying to identify all the chaperones involved in the replication of respiratory syncytial virus, which is the most important respiratory infection in infants. And the second project that we have is trying to address the fact that we know that RNA viruses have very high mutation rates, and we know that most mutations are deleterious to protein function. And so we're trying to understand how can RNA viruses deal with having such high mutation rates. And so for this, we are experimentally putting in all possible amino acid mutations in a model viral protein, and then trying to understand how the virus is able to accommodate these mutations. And from this, we hope to understand some principles underlying protein design that can be used to synthesize more stable or more robust proteins.
Universitat de València.